Grow, Grow?
Yeah. We're live now, by the way.
Yeah. Okay, great. Let's go ahead then.
Okay, perfect. Hello, everyone. I'm Ash from the Neurizons organizers. And before I introduce our next speaker, I'd like to remind everyone that you can ask questions with an Ask a Question feature. And please don't forget to indicate with a plus or a minus whether you want to come on screen to ask your question. Please don't forget that this event is being recorded. So by agreeing to come on screen, you're also consenting to being recorded. Additionally, you can also rewatch the talks later on Crowdcast for up to a week after the conference in case you've missed anything. So our speaker now is Professor Ilona Kadow. She's a professor at the Technical University of Munich, and she studies how the external environment and the internal environment of an organism modulate its behavior. More specifically, she studies questions such as differences in perception between starved and fed individuals and the mechanisms underlying their different choices, and how computation at the neuronal and neural level are affected by different states and experiences. Her group studies these questions in the context of chemosensation and chemoperception in Drosophila and addresses them at levels spanning from genes to behavior by employing a variety of approaches from behavioral analysis to optogenetics and in vivo multiphoton microscopy. So without further ado, please give a virtual round of applause for Professor Grundwald Kadow.
Hi, thanks Ash for this very nice introduction, and thanks for having me. I hope everyone can see and hear me all right. I actually don't see you, which is weird, but I'll just go ahead anyways. So essentially my lab's been interested, or I've been interested in understanding how our brain helps us to survive, essentially. So to me, the main purpose of a brain is to help the organism survive. And in order to do so, what the brain needs to do, it needs to keep track of not only what's going on around us, but also what's going on inside of us. And that's a very difficult task. And because the task is so difficult, we chose to work on this creature that you see right in front of you, which is usually not blue and brown, but usually more brown and red. Drosophila melanogaster, that I'm sure many of you are familiar with, because it offers us many opportunities that potentially more interesting animals or humans, such as humans, would not offer. But I hope I convince you of that during my talk. So if we think of behavior, behavior is incredibly complex, and very often in laboratory assays, we try to really make this very simple. We think of, go or no go. Do we observe something or do we not observe something? But if we look at behavior more carefully here, this cheetah running after his dinner, presumably, we can appreciate that this animal needs to make a lot of decisions, both of them actually. Of whether to continue running, of whether or not to stop, and what strategy to do next, etc., etc. So, and as this is constantly changing, this is a very dynamic thing the animal is doing. The brain needs to constantly update the perception and also the motivation and goals. And so if you think of this, the way this animal is probably going to think of this is, look, I'm very motivated because I'm hungry, and I'm going to put a lot of effort in because I'm hungry, because I expect the reward to be great. And so essentially that's the theory behind a lot of decision making, is that our internal state and our need, our motivation regulates how much effort we're willing to put in for an expected reward. And so the question I'm very interested in, and that I want to discuss with you today a little bit, is how are behaviors prioritized, maintained, or suppressed depending on need and motivation? And as I told you earlier, the brain needs to integrate a lot of information to make this kind of decision. And so obviously there is our sensory world around us that the brain needs to process. And I think we're starting to know more and more about how that works. But then there's this other component, and that's our internal state. And by internal state, I mean metabolic states, physiological states, but also behavioral states. And so I'll discuss all of this a little bit during this talk. And all of that needs to be integrated for the brain to make a useful, decision for the organism. And of course, any sort of behavioral outcome will then again have an impact on how we perceive the world around us and so on. So it's a constant loop between the perception of the world around us, inside of us, and the behavior it elicits. And in my lab, we have chosen to study this, in particular on the example of olfaction, because olfaction tends to be one of the senses that is highly influenced by our states, such as hunger states, for instance. Usually we perceive food smells as much more positive when we're hungry and not when we're not hungry, for instance, just as a simple example. And also because they're very important for most animal species and highly conserved, for instance, for finding food, avoiding danger, and finding mating partners, and so on. So as I told you, we do this in a fly, and the big advantage in a fly is that, first of all, flies do make a lot of decisions that are similar to our daily decisions, of course, at a different level, but still they need to find food, mating partners, avoid dangers, etc. Or in short, they need to survive and they need to reproduce. And working in flies allows us to make, to make the link between genes, neurons, and behavior. And a little side note, what I kind of like about it, there is no consideration, or not much at least, regarding the ethics. We don't need specific animal protocols, so we're very free in what we can do and how we can do it. As I mentioned earlier, and before I want to go into our specific work, I just want to tell you, a brief introduction of what we know already about the olfactory system in the fly. Well, one thing we do know, that there is a high degree of conservation between different animals, and that has been written about in many review articles. And so one thing is clear, there is external odor information that is being brought through olfactory sensory neurons that in us sit on in our nose epithelium, and in the fly sit on its antenna. And then from here, the information is brought to the antennal lobe. In us, that's called the olfactory bulb. And then from there, the information is transferred to two main, at least we know of two main, but there's potentially more, higher brain centers. One is called the mushroom body, and one is called the lateral horn, and just basically for the physical appearance. And essentially what is important is that the information that comes from the antennal lobe is split into parallel pathways, processed probably somewhat differentially, and then basically in order to produce behavior, this information needs to then come back together. And we assume that the information coming from these parallel pathways then together will give rise to a behavior. Work of many labs over many years have suggested that the mushroom body is primarily responsible for the ability of flies to learn or to be able to associate odors with, for instance, electric shock, whereas the lateral horn is thought to be responsible for innate behaviors because the neurons in the lateral horn show by and large stereotypic responses to odors. We also know through work of many labs, including ours, that the mushroom body is not only required for associated memory formation, but it's also required to allow flexibility on any kind of decision. So basically, to be able to adapt a response to an odorant to a particular state, such as hunger states or a particular context. And that's something I want to talk about for the rest of the talk. As a side note, one of the beauties of the fly system is that since a couple of years, and this is an ongoing effort by many labs that are collaborating, mainly driven by Janelia and people in the UK, is that we now have the adult fly connectome. That means these 100,000 neurons that exist in the adult fly brain, we're starting to get an idea how they connected, who speaks with whom. And one of the things that we realized is that the brain is incredibly interconnected. It's an interconnected network of many recurrent connections, and some of them are long-range recurrent connections. And that puts the brain somewhat in a different way of thinking, or us in a different mindset, because that really means we need to consider the brain as a whole and stop thinking of it as individual regions or very sort of unidirectional pathways. And that fits very well to what I meant earlier, that it's a constant loop between inside and outside and behavior. And it seems to be that this is exactly also what the brain needs to do. So let's get back to the question I posed earlier. And the question was, if you remember, how are behaviors prioritized, maintained, or suppressed, depending on need and motivation? And of course, cheetahs would be great, but as I said, we do it with flies. And so we devised an assay where we essentially put a little fly in a ball and let it run happily. Well, I believe it's quite happy, or, you know, at least it looks quite happy. It's running. And we can stimulate it from the front with odor and or other things. We can also do optogenetics, as I will show you later. But essentially, we can stimulate it in a very precise way with a stimulus. And so we did this to mimic foraging behavior in a fly and to be able to analyze this foraging behavior in a high level of detail. And so we do that. What essentially we did, we were actually pretty nasty to the flies. We stimulated them 10 times with a positive odorant. In this case, it's vinegar. Flies love that. They run after that. And we did this 10 times, but we never rewarded it. And we wondered what would happen. And so if you average 10 trials of many, many flies, what you see is that the flies initially, once you put the odor on, the flies start to run very fast after the odorant. You can see this here. And then as soon as the odorant is off, they actually stop, they start turning, and then at some point they spend time running again, presumably in search of deodorant. But the more interesting thing that we noticed comes when we looked at the single trials instead of averaging everything. So we noticed that, so in blue here, this is the first trial, and in orange, that's the 10th trial of a fly or of many flies. And what you can see is that flies run longer and faster at every unrewarded trial. And you can see this here. This is not only dependent on, so these are the trials. This is trial 1 and trial 10 for a not starved fat animal versus a 24-hour starved animal or a 48-hour starved animal. And I think what you can see is that the effort or the time of spent running after the odorant of a fat animal is very small as compared to the 24 or 48-hour starved animal. And not only do they run more on average, they also show this quite drastic increase in running. So this suggests that flies, hungry flies, are very persistent in getting to a potential food source, and that not getting a reward at every trial actually leads to quite the opposite. Instead of giving up, they basically try harder. So we wondered, what keeps animals going without success? And I think that's not something that just flies experience in our laboratory. I think that's something we experience all the time. And I think it's, for those of us who have kids, for instance, it's something very important to consider is like, what do you do? How can you keep yourself and anybody else going, even if it doesn't work out right away? And so how do we do this carrot thing? And so, as you have seen, the behavior evolved over time. So from trial 1, where the animal was running a little bit after deodorant, to trial 10, where the animal was running a lot more after deodorant, we figured this might be some sort of working memory or some sort of learning. And so in a fly, conveniently, we know a lot about this at the cellular level. And so I'm just going to review this very quickly for you. As you probably all know, sensory information in a, pretty classical way, if we think about learning, is received by a neuron. This neuron is then connected, presumably, to an output neuron, and this neuron then is giving output to a particular behavior. In addition, during learning, what happens, there is a modulatory neuron, and this modulatory neuron is connected to one or both of these neurons, the neuron and the output neuron, and modulates the connection between these two neurons in a way that modulates the output. So, for instance, it can mediate reward or punishment. This then will change the connection or the synaptic strength between the neuron and the output neuron, and this will then lead to a difference in how sensory information leads to behavior. So conveniently, in the fly mushroom body that I mentioned to you earlier, the center where learning and memory takes place, we have exactly this type of beautiful structure. We have Kenyan cells, those are the ones that receive, in particular, olfactory information, and these project those to the mushroom body so-called mushroom body output neurons very creatively. And then the modulatory neurons, in this case, are dopaminergic neurons, as you know, very often also in our brain. And these receive information about reward or punishment and then modulate the connection between the Kenyan cell and the mushroom body output neurons. Luckily, through the connectome that I mentioned earlier, we now know that this is not just a one-way street. This is a highly recurrent circuit. And in the mushroom body, we have about 15 interconnected compartments full of these modules, of highly interconnected modules with recurrent circuits. And these are obviously perfect to allow this type of learning. And so we ask, could they also, could this module be able to produce the behavior we're seeing, basically this persistence and this increase over trials? And if we do this, we basically got together with Juliana Georgieva and her postdoc Marina Wozniak and asked them, could this module that we see in the fly mushroom body actually reproduce behavior? And I want to go through this very quickly, but essentially, if you compare in the last graph, if you compare the data with our fitted model, you can see that it's perfect. So this little module alone is able to potentially reproduce this behavior. And so then we ask, is that really what's going on? And so to do so, we can now go back and use fly genetics and ask specifically which of these neurons, the Kenyan cells, the dopaminergic neurons, or the mushroom body output neuron, are indeed involved. And we can do this by activating or inactivating them. And we did this in many ways, and I'm just going to show you a couple of them because of time. But essentially, we first ask, is dopamine required for the persistence? And here, conveniently in the fly, we have dopaminergic neurons, as I said, in the projecting to the mushroom body in this little module that I showed you earlier. And through work of a lot of labs, we now know that there is a cluster of them called PAM that seems to be primarily responsive for responding or for modulating information in the context of positive, like sugar stimuli. And by contrast, there is a negative context, and that's the PPL1 neurons that are required, for instance, for electric shock learning. And vice versa, if you activate using, for instance, optogenetics, these PPL1 neurons, this basically mimics electric shock. And so we inactivated these neurons during the task. And basically what we see, if you look at the control animals, what we've seen before, these are 24-hour starved animals. You can see that between trial 1 and trial 10, the animal runs faster, as we've seen before. So now if we inactivate the PAM, so these positive context neurons, we see absolutely no difference. However, if we do the same with the negative context neurons, the ones that can mimic electric shock, we basically see that the learning curve or the change in behavior and the persistence of the fly is severely reduced. And this suggests that basically some potentially negative context or negative feedback is driving the fly when it doesn't get rewarded. So we can now next ask, where and how does dopamine act? And I told you before that dopaminergic neurons could modulate either the Kenyan cells or the mushroom body output neurons. And in fact, there's also not only potential target neurons, there's also potential differences in receptors. And so as you may appreciate also for humans or other animals, dopamine activates different receptors. And for instance, in the fly, we have dopamine receptor 1 and dopamine receptor 2, and these use slightly different G protein signaling and have different functions in the brain. What we can now do by targeted knockdown with RNAi, we can basically knock down these dopamine receptors in specific sets of these canyon cells in these different compartments, and we can basically then say, ask whether this is required. And if we do this and we knock down actually dopamine receptor R2, what we find is that if we knock it down in a specific subsets of these canyon cells, we can basically completely mimic the behavior that we see when we inactivate dopamine signaling from these PPL neurons, suggesting that these neurons target or work primarily on a small subset of these olfactory stimulus receiving neurons. And of course, we then ask if there is canyon cells that receive this information and dopaminergic neurons that modulate this information. There must also be a specific neuron that then receives this information and that passes it on to the next, to basically direct behavior. And this is also, again, what we see in another genetic screen where we're basically looking for which of the downstream neurons could be responsible for changing this behavior. And again, we use genetics. And so if we inactivate this specific mushroom body output neuron called MVP2 or gamma-1 peduncle projecting to alpha beta, doesn't matter, that's not important. But essentially what we see, if we inactivate it in a starved fly, we see basically the behavior that we see when we inactivate signaling from dopaminergic neurons or dopamine receptor signaling in a specific subset of canyon cells, suggesting that these are really the important outputs. And in addition, if we activate these neurons in the fat animal, we can basically, not completely, but we can basically mimic the starved state, suggesting that the modulation of these neurons, and this is really just, as you can see here, this one little neuron here, that this neuron is basically the main neuron that is modulated during this persistence behavior. But of course, as I mentioned earlier, animals, when they run after a prey, for instance, they don't only have to make the decision of how long they keep going, but they also have to decide when to stop. And in particular, they have to stop when they've reached their goal. And this seems like a trivial thing, but one thing one needs to think about it is that, of course, some of the sensory information that made the animal run after the animal in the first place, after the prey in the first place, the olfactory stimulus, for instance, is still present when the animal has reached a goal. So there will be basically multiple sensory information coming in, and this needs to guide whether the animal keeps going or actually stops and considers feeding. And so we wondered how this could be, and we figured there must be neurons that basically override or stop or inhibit this olfactory signal that leads to running in a way that basically makes the animal stop and then start to eat. And so these neurons are presumably responding to food or feeding related cues. They're presumably connecting between feeding or taste centers to olfactory processing, and they're likely involved in motivation-dependent modulation. And so a good candidate for this was octopaminergic neurons. So octopaminergic neurons in a fly resemble in several aspects, and not in all, obviously, the norepinephric neurons in the vertebrate brain. And so now, to cut a long story short, we identified two neurons. One is called octopamine neuron VPM3 and the other one VPM4. And if we modulate them, so using optogenetics, for instance, we can activate them. If we express crimson in this case, which is a red-shifted channel to activate neurons, what we basically see is that we see this. And so essentially, in the presence of the odor stimulus, we can activate these neurons. And if we do this, we can completely suppress the fly's running behavior, suggesting that this neuron is sufficient to override the go signal that the animal receives through the odor signal. And basically, we also know that these neurons are responsive to food-related cues. So now we can even go a step further, and that's what fly work allows us to do. We can now ask, is there really a connectivity? How does this work at the synaptic level? And so here again, we can turn to, in collaboration with Davy Buck and Greg Jeffers, we can now ask, these neurons are all involved. I showed you this mushroom body output neuron, MVP2, and these other two octopaminergic neurons are involved, but what do they have to do with one another? And if we use now the connectome, we can actually trace the potential connections between, and what we find is that these neurons are indeed directly synaptically connected. So there is a direct connection from these neurons that sit in the taste center of the fly, or basically I like to also call it the brainstem of the fly, all the way up to this higher olfactory center, and they directly make a synaptic connection. And I don't have time to show you the data, but essentially using other types of assays, including in vivo imaging, we were able to basically dissect an entire circuitry down to very specific types of neurons, or even single neurons, and particular signaling molecules that is responsible for this complex behavior that I showed you. So essentially, if the fly is not successful, so it's not being rewarded for an odor cue, so the fly receives an odor cue and runs after it, it's not being rewarded, then it appears that dopamine receptor 2 signaling basically keeps this loop going, and that through, and I didn't show you the data for this either, through a connection to the lateral horn, increases odor tracking and leads to persistent food odor tracking. By contrast, as soon as the fly is successful, these neurons, these octopaminergic VPM4 neurons, are being activated. This then, through a direct synapse into this olfactory circuit, acts basically what we think acts like a brake, and this then stops the circuit and stops the odor tracking. And so to put this together, we believe that these two neuromodulators, dopamine and octopamine, through direct synapses on the same subsets of neurons, counteract each other and thereby toggle behavioral persistence versus basically the ability of the animal to change its behavior, and in this case, to stop and start feeding. So, of course, as every work that you do, it triggers a lot of new questions, and you may have questions for this. But we also, of course, had a lot of questions. And one of the questions was, so if dopamine really is telling the animal, for instance, that it's not being successful, how does it do this? And given that we know in not just the fly's brain, but in any of the brains that people study, there seems to be many different types of dopaminergic neurons. And what is also interesting, these different types of dopaminergic neurons have been implicated in all kinds of behaviors. Some study them in the context of motor behavior, if you think of Parkinson's disease. Others study them in the context of addiction, others in the context of valence perception, error prediction, learning, etc. So a lot of information is apparently being predicted, or basically being passed on through dopaminergic signaling to other parts of the brain. And so if we go back to our model, the insect mushroom body, we're in a lucky situation that basically there's only 20 different types of dopaminergic neurons that innervate the mushroom body of the fly. That comes down to about 130 cells, and we have a very good understanding of their anatomy. They're organized in 15 interconnected compartments. And they're, what we know is they're definitely, and I mentioned this earlier, required for aversive and appetitive associative learning. And that has been shown by many, many labs, such as the Waddell lab, the Tully lab, the Heisenberg's labs, the Tanimoto labs, et cetera, et cetera. So many labs, too many to name them all. But essentially, what we do notice is that it's probably that these neurons can do more than just tell whether there is a positive, whether there is a shock around or a sugar around. And so we wondered whether these neurons as a population, what kind of information are they able to pass on to this higher brain center, such that the fly is able to make this type of flexible decisions that it needs to make in such an assay where it constantly needs to update its representation of, for instance, an odor cue. And so we decided to, instead of what we've done in the study that I showed you earlier, where we looked at single components and tried to manipulate individual components of the network, we decided, let's look at the whole of these neurons in the network, and let's try to understand whether there are specific subtypes and how they talk to each other, and whether there's differences in how what communication, what information they give to the higher brain center. And so I would like to briefly go back again to this anatomy of this center. And so as I told you, there's two main subgroups, the PAM dopaminergic neurons and the PPL dopaminergic neurons. And these then innervate the mushroom body in these 15 different compartments that come in these different names, alpha 3, alpha 2, etc. That doesn't really matter unless you're a fly person. But what matters is that there's different compartments that are being differentially innervated by these two subsets of neurons. But primarily, there is this distinction between the neurons, the compartments that are innervated by the PAM neurons that seem to be more responsive to positive contacts, and ones that are innervating the other subtype, the other horizontal branch, vertical branch, excuse me, of the mushroom body that seem to be more responsive to electric shock. But again, there's 15 different compartments, and we do know that they are likely not only responding to electric shock and sugar. However, they probably, and there's work by other labs. I want to, in particular, highlight the work by the Ruta lab, but also work by the Waddell lab and others that showed, yes, of course, these neurons can do a lot more than just that. But then the question is, how do they do this, and how do they share the work? And so in order to look at this, we decided to basically do a sort of population imaging, where instead of imaging individual cells, we would image them all in the living animal and try to find out what these neurons respond to and how they may encode information. And so we basically image using 2-photon imaging the entire region of the mushroom body and all of the dopaminergic neurons. And then a challenge, and we solved this challenge together with the Berlin student and Ruben Portuguese lab to get these information from different fly brains onto a common substrate in a form of basically this big data. And essentially, he developed a way so that allows us to basically register the information from different fly brains in 3D. And then flatten it out so that we can look at it, look at it in a 2D way, and basically can look at the different signals of the compartments. And so if we do this, we first started to ask, yeah?
Professor Kadow, I'm gonna have to ask you to wrap up in about a minute or two. Sorry to interrupt.
Oh, okay. Gee, now I need to rush. Okay, great. Thanks for letting me know. Yeah. So essentially, so the first question we ask is, do dopaminergic neurons actually predict odor identity? And if you look at these different responses, you can see that there is indeed, they look slightly different. And so this is about data of about 400 flies or so. But if you look at the different responses in this PCA analysis, you basically have the impression they're all clustered. However, if you look a bit more careful, you can see that there is a difference between how, for instance, this odor, which is a pheromone, or this odor, which is a food odor, activates the dopaminergic neurons. And if you look at this, you can also see that there's some predictive power in whether the animal, in just looking at the response of the dopaminergic neurons, or whether the animal is experiencing a food odor or pheromone or a highly aversive odorant. And so basically, when we calculate this, the population activity of these dopaminergic neurons is about 3 times better in predicting the identity of the odor as opposed to chance. But more importantly, this also suggested that maybe identity is not the main thing that the dopaminergic neurons tell the brain, but instead it's probably something to do with valence. And so this is exactly the case. If we looked at the different compartments and we ordered the odors in negative and positive valence, we basically saw that some regions were more responsive to positive valence, whereas others were more responsive to negative valence. And if we do a logistic regression, we can very clearly see that the neurons, the population activity of these dopaminergic neurons as compared to chance is able to predict by 70% confidence whether the fly is experiencing a positive or negative odorant. And similarly, we did the same for a positive and negative taste. And here we used a slightly different method of microscopy called the light field microscopy, which allows us to do whole brain imaging. This is a method that has been developed by my postdoc Sophie Aymon in the lab of Ralph Greenspan. And I don't have time to go into it, but essentially what it allows us to do is to image whole brains in a very, very fast time resolution. And so when we now stimulate the fly with a positive or a negative taste, we can also find that there are certain compartments that are more responsive to negative taste, whereas others are more responsive to the positive taste. And so we can clearly see that, again, if we look at a regression coefficient, that some of the compartments are highly correlated with a positive or negative response. This dopaminergic neuron activity is also subject to metabolic state. So if we now compare all odor responses across these almost 400 animals that we looked at between fed and starved animals, what we can again see is that by just looking at the responses of the dopaminergic neuron, we can predict with about 65% certainty of whether the animal was hungry that perceived the odorant or whether it was not. So it seems that just in these dopaminergic neurons, there's a lot of information to just basically about valence, but also about metabolic state. However, we also noticed, given that animals move around a lot when they do any type of behavior, and this was particularly the case for the assays that we've been using, and of course we do know from work in other animal models that dopaminergic neurons are responsive to movement, we wondered if also the neurons in the mushroom body of the fly are responsive to movement. And so basically, when Sophie recorded, again, using whole brain imaging, and she just recorded from the dopaminergic neurons, you will see as the animal started to run, you can see that there is a very clear increase in activity. And this is also very clear if you look at this. So here, this is every time the animal moves, and the movement is walking and not grooming, for instance, we see very clearly that there is a response and activity. And this is not true for all compartments, but that is true for several compartments. And we can see this here, that in particular, these compartments seem to show high activity with walking. And perhaps equally interesting, if we just look at the whole brain activity between a fly that is resting or grooming or a fly that is walking, we see that the activity of the brain as a whole changes dramatically, or the state of the brain, so to speak, changes dramatically whether the animal is resting or grooming or whether the animal is walking, suggesting that there's probably a lot that walking activity does to the perception, not only in dopaminergic neurons, but in general. But to wrap this up, basically, what we find is that the dopaminergic neuron population that innervates the learning center in the fly basically shows responses to or encodes all kinds of relevant information as the fly is doing a behavioral task. And these are, for instance, the valence of sensory information. Odor and taste is what we've looked at. And I hope you can appreciate, so in blue is everything that's more correlated to negative valence, and in red everything is more correlated to positive valence. You can see that this valence representation, of course, is not completely the same for these two sensory modalities, but you can hopefully see that it is highly similar, and that probably the valence representation in dopaminergic neurons is less dependent on the sensory modality, but really the valence. We also see a very clear modulation through starvation, and I haven't shown you the data. We also see modulation through the mating state of the animal. And last but not least, we see a very strong modulation through walking activity. And so this suggests that these neurons seem to integrate a lot of the contextual information, from internal information to state information, all the way to sensory information, and that this integrated information that is represented in this population of dopaminergic neurons is probably really important for the fly's perception and also for its behavior. Before I wrap up, one thing that we've also noticed, and we're looking into this now, is that I've now shown you average data, and this is basically average data from 400 flies. But if we look at individual flies, we do see that despite showing overlaps in representation for a particular odorant, we also see strong variation between individuals. And so this is one of the things that we're currently tackling is why is it that different flies respond differentially? As a group, they respond similarly, but still there's individual differences. Is that because of stochastic differences in development, or is that because of experience, or a combination of the two? And I think this will be something that will be very interesting to try to figure out. And I will skip this in the interest of time. But essentially, our goal for the next, couple years or so will be to use a lot of whole brain imaging, so to basically, or population imaging of certain brain regions, and then to try to use this whole brain activity to detect in which state the brain is. Also use this to derive functional connectomes between brain areas that are correlated in a certain behavior, and compare them to the whole brain network that we're now getting through the EM connectome, and then use this information to derive models that basically represent behavior in the dynamic process that it really is, and not just in a very sort of rigid state with single stimuli. And so I'd like to very briefly acknowledge people. So this is my lab, more or less. It's always changing a little bit, but I'm very grateful to my three postdocs, Jeff, Siju, and Sophie, who did different parts of the work. And also to my former student, Serjan, who was a driving force in the first study I told you, and Siju was the driving force together with Sophie in the second study I told you about. And I'm very sorry to be over time. It's a bit difficult to follow this because I only see my screen, but thanks anyways for listening, and I'm looking forward to receiving your questions. Thank you.
Thank you, Dr. Kadow. So I think we don't have a lot of time for questions, but we'll still try to take one or two. The first one is from Vasil, and he asks, for how long will the hungry fly run after the appetitive smell without receiving a reward? Does it habituate over time and decrease performance?
If they you mean like how long does it take for a fly to basically give up?
How long does it take for it to habituate
to running on the ball? For how long will the hungry fly? Is that the first one you were? Yeah. So okay. So this is essentially it's a difficult. It seems like a very easy question to ask, but it's not so easy, because if the flies are hungry and we do let them run, and you've seen this already in one of the slides I showed you, is that essentially basically you do see that at some point it goes down a little bit, and they do give up. But the problem is that, do they give up because they're physically exhausted, or do they give up because they have changed the representation of the stimulus? And so that's been very difficult. We've done these experiments, but we have a hard time to interpret them. And so what we are doing right now is we're hoping by doing more of the imaging as the animal is doing these things. And unfortunately, these experiments are very, very difficult to do, because the problem is that if you look in a fly that is under a microscope supposed to do something is not the most happy fly. And so it's a very difficult prep. And so it's not an easy question to ask. And so the short answer is we don't know. It does give up eventually, but we're still not sure why it gives up. But we're on that.
He's also asking if this is reflected.
Yeah, so good question. So we're also looking in it. So more broadly speaking, we do know that the activity of the dopaminergic neurons does change over time. We do know that it is required for flies to continue or to give up. However, we still have no sense of what it is that makes this. Why does this experience seem to activate or require neurons that are usually, or for instance, also activated by electric shock? And so one thing we're very curious of trying to figure out is, what is the representation of negative experience, and how can basically a failure to obtain a goal, why is it perceived in a very similar manner as an electric shock? And so we're very curious to find out about this, and this will require a lot more imaging as the animal is doing these tasks.
Okay, so I think this will have to be the last question, and I guess you can kind of field the rest of the questions in the comments later. But the next question is from Yasmina, and she says, very fascinating data. I was wondering, is it possible to stimulate OA VPM4 neurons in the absence of food in order to mimic depression?
Yeah, so basically what we do is we activate the neurons in the absence of food. We can also stimulate the flies with food, but basically we at the moment, it seems like if we activate them, this seems to basically be sufficient in the absence of food to completely suppress running behavior. So we think that these neurons are basically a connection between feeding or feeding-related behavior and olfaction. And so essentially, as these neurons are being activated, even in the absence of food, that's sufficient to suppress basically the activity in the olfactory circuitry, and essentially that stops the flies from running. Yeah, so we think they're sufficient.
Okay, so with that, I'd like to thank you for your wonderful talk, and perhaps you'd be able to answer some more of the questions, because there are quite a few interesting questions that have been asked.
Okay, great.
The chat.
Thank you.